Hi, this is Mr. Worski here, and today we will start molecular genetics of eukaryotes, which should go on the heels of molecular genetics of prokaryotes. Let's talk about eukaryotes, shall we? Great. So, if you have any questions, don't forget you can pause the video, you can check your textbook, you can review the video again, and you can ask me questions tomorrow in class. So we started off with DNA and the amount of DNA in prokaryotes last time. We were talking about molecular biology of cells, and the DNA of the cell is different in different species for eukaryotes. What do you mean? Well, Drosophila has 2 times 10 to the 8th base pairs per cell. For you people, we have 7 times 10 to the 9th base pairs per cell, but we do not have as much DNA as salamanders do. Now remember, in eukaryotes, not all the DNA is used to produce proteins. In fact, there are different classes of DNA. So you might be asking yourself, what are the different classes of DNA? Awesome question. The first class are short, tandemly repetitive DNA sequences, which are also known as STRs. These are short, anywhere from 5 to 10 bases long, and they're repeated over and over and over again. So about 10 to 15% of our DNA, STRs, telomeres, centromeres, there are specific places on chromosomes, but they can vary in length. And keep that in mind because the next series of videos, we're going to mention this again. STRs. The second class are interspersed repetitive DNA, or IRs. They're longer than STRs, anywhere from 150 to 300 bases. That varies too. They are similar but not identical to each other. They are scattered throughout the genome. Some have known functions. For example, though 20 to 40% of our DNA are IRs, sometimes they can produce histone proteins or rRNA genes. The ALU gene is anywhere from 180 to 200 bases. It is an rRNA gene with no promoter, and it is repeated about 1 million times in our genome. And hopefully we'll be able to manipulate and multiply that in our PCR lab. The line 1 is anywhere from 1,000 to 6,000 bases long. It is the gene for reverse transcriptase, and it is 100,000 times in our genome. Every time we get one of those retroviruses, we get the line 1 gene. And then finally, the single copy gene. That should be the number 3 right there. The single copy gene is our genes, anywhere from 45 to 65% of our genome. These code for proteins, I should say proteins, but only 3% is used at any one time. So our DNA is packaged as a eukaryotic chromosome. So we're going to have to add this to our DNA discussion. So each chromosome is about 2 times 10 to the 8th DNA base pairs. They are 6 centimeters long. Each cell has about 2 meters of chromosomes per cell. There's 25 billion kilometers of DNA per body. And they must be packaged as chromatin. And you remember that is DNA plus histones. Now, there are 5 types of histones, and they have been cleverly named as H1, H2A, H2B, H3, and H4. The packaging unit of a histone is called a nucleosome, and they're made up of 8 histones, 2 copies of H2A, 2 copies of H2B, 2 copies of H3, and 2 copies of H4. All together that form one ball that DNA wraps around. You might be asking yourself, where's H1? And they're attached from nucleosome to nucleosome on top, and it keeps everything nice and stable. There are 146 bases around each nucleosome and 30 to 60 DNA bases between the nucleosomes. And this makes up chromatin, and these are how our chromosomes are packaged. We have pictures of the different proteins here, and we have the nucleosomes there. The DNA is wrapped around, there's 146 DNA bases wrapped around here. There will be 30 to 60 DNA bases between, 30 to 60 here. And the H1 histones are linked to each one of the nucleosomes and keeps everything nice and stiff. See right here is also nucleosome, all the different proteins together. And we have the DNA bases around here, and it's about 146 DNA bases wrapped around here, 30 to 60 in here, 30 to 60 there. So we talked about how the DNA is replicated in prokaryotes, and we're going to talk about it here too. DNA synthesis happens at many points on the chromosome, and it happens bidirectionally. It happens about 45 bases per second. It's a little slower because the DNA must unwrap from the nucleosome. So we talked about the operon, or how the genes were organized in the prokaryote. We're going to talk about it now here. There is a promoter with a TATA box, about 25 to 80 bases upstream from the start site. And then we have RNA polymerase binds to the box. Enhancers are located thousands of bases away on the enhancer. So here we go. We have the enhancer here. I have a space of DNA here. The TATA is here, and then we have the promoter right here. Then the genes are here. This needs a promoter right in there. Okay. So transcription in eukaryotes is similar to transcription in prokaryotes, except there's only one gene per promoter, where in prokaryotes you could have many genes per promoter. There are three types of RNA polymerase. We have RNA polymerase 1, which transcribes the large rRNA subunit. RNA polymerase 2, which transcribes the genes and also vDNA. And RNA polymerase 3, which transcribes a small rRNA as well as the tRNA genes. So remember, in eukaryotes, the original transcript is called hnRNA, and it must be modified and edited to become mRNA. Now, the DNA must be unwrapped from the nucleosomes during transcription, so that takes a little bit longer too. So it's a little slower than prokaryotic transcription. So how do we control eukaryotic gene expression? Well, in prokes, we controlled it with an operon, and that was simple. There was pressable, there's inducible. We're good. Maybe we have some crap camp going on there to increase transcription. However, control for eukaryotes can happen at different points of protein synthesis. So if you control any one of those points of protein synthesis, you will control gene expression. So the first way to control gene expression is to control transcription. So if we stop transcription, the protein is not produced. If we increase transcription, more protein is produced. How can we control transcription? Oh, there are a bunch of different ways to control transcription. Boo. In prokaryotes, there's an operon. Now here we can condense the chromosomes. Condensed portions of the chromosomes are not transcribed. Relaxed parts of the chromosome are transcribed. Condensed parts of the chromosomes are called heterochromatin, where the relaxed chromosomes are called euchromatin. Methylation of DNA. So after synthesis, an enzyme will add methyl groups to cytosines in inactive genes. The more methyl groups that are attached to the cytosines, the less likely they are to be transcribed. Now, interestingly enough, when your first fertilized cell, when the cells start to divide, at the third division, methyl groups are added to turn off genes. So gene regulation is seen way back then. So we have methylase adds the methyl group to the cytosine here, right? And we see the methyl, and the more methyl groups you have, the least likely it is to be transcribed. So there are things in your genome called HERVs, or human endogenous retroviruses. So after virus, the vDNA that's in your DNA is methylated so that you don't have the virus produced all the time. So some viruses can escape methylation and start reproducing again. And then your body has to attack those things. And they think that HERVs may be active in people with schizophrenia, multiple sclerosis, and Parkinson's. Histone acetylation is when you add an acetyl group to the histone proteins, and it loosens the grip of the histones on the DNA, and that can actually increase transcription. So we have a picture of it back here, in which we have the histones are attached to the nucleosomes here, and that increases transcription. So we can decrease transcription and increase transcription. Now, methyl groups and acetyl groups can be controlled by the environment. So as you grow older and exposed to different things, you can turn on and off different genes. This is why identical twins aren't identical, because they produce different proteins due to methylation and acetylation due to different environmental exposures. This whole study of how the environment affects gene expression is called epigenetics. So we have acetylation here, acetyl groups. You notice that the DNA between the chromosomes, the nucleosomes gets larger, so they can produce more mRNA, or hnRNA, which is converted to mRNA. How else can we control transcription? Transcription factors. These land on the promoter and enhancer, and it forms a loop. So here's the enhancer, here's the promoter, here are the transcription factors, right here. And this loop will increase transcription by attracting RNA polymerase. So if the promoter and enhancers are similar, you can turn on groups of genes that have similar functions at the same time. So there are over 100 different transcription factors. And these genes code, and they're Hox genes that code for the transcription factors, and they turn on groups of genes at the same time. Hox boxes, because they similar transcription factors land, turn on all these genes. You can also regulate the specific binding proteins that bind to DNA, and they will stop RNA polymerase, so they stop transcription. Sixth way to control transcription, and we're only talking about transcription again, steroids. What they can do is they can pass through the cell membrane, they pass through the nuclear membrane, they bind to a protein, and then that complex binds to the promoter and acts as a transcription factor. So steroids just increase transcription. So far, we've learned about 2 ways to increase transcription: acetylation as well as steroids. Everything else that we've talked about, stop transcription. 7, inhibit RNA metabolism. How do you do that? Well, you have a protein or a molecule binding to RNA polymerase, and that will change the shape of RNA polymerase so that it cannot transcribe and produce hnRNA. So what happens if hnRNA is produced? Well, you can control the post-transcriptional process to control gene expression. So you have hnRNA, and you need to change it to mRNA. If it's not changed to mRNA, you will not have a protein produced. How do you produce mRNA? Well, you need to modify the ends. So the 5' end is not modified by the methylguanine. If the poly A tail is not added to the 3' end, and if the introns are not spliced out, you will not have the production of mRNA. And if there is no mRNA, it will not leave the nucleus, and it will not produce a protein. So those things have to happen. In addition, there is a mRNA-P, which actually escorts the mRNA to the nuclear pores. If that's not done, it will not encounter a ribosome. It will not produce proteins. So those things have to happen after the hnRNA is produced. First, you have to produce mRNA, and then you have to escort the mRNA to the nuclear pores. Again, if that's not done, no protein, because no RNA or ribosomes are encountered. We're going to stop there, and I'll see you in the next section. Have a good night.